So welcome everybody to our 37th webinar. I hadn't realized we'd had so many and time flies, but thank you for joining. My name's Keith Goulding. I'm chairing this webinar, and all of the previous webinars are on YouTube. So you can catch up very quickly just by clicking on the website and looking at previous webinars. There might be several that interest you, so do have a look. Ben Sykes, our Executive Director, will be organising the technology and making sure everything works, stopping and starting the talks. So just to introduce our two speakers today, first of all, Andy Gregory. Andy is the lead of the Rothamsted Long-Term Experiments National Bioscience Research Infrastructure, as it's now called, which includes Broadbalk and Park Grass. And Andy coordinates the maintenance and development of these, the world's longest running agricultural field experiments, and the associated sample archive and the data sets to support new science in sustainable agriculture, food security, and climate change. Andy is a soil scientist with research interests in soil physical properties and soil carbon, and how these are affected by the way in which soils are managed and how they impact on other soil functions. And then we've got two speakers. So Jon Storkey, who you might have heard before. Jon is a plant ecologist who studies the impact of management and environment on plant communities found in farmed landscapes, with the aim of balancing their contribution to biodiversity with their negative impacts on crop yields. And the long-term experiments at Rothamsted have been a valuable resource for Jon's research, and he's worked on this for many years. Please, when the talk starts, turn off your cameras and microphones if you haven't already. I see most of you have, so thank you for that. If you have a question, please use the chat facility, which is the little balloon type symbol down in the bottom right-hand corner. Click on that and you'll see all of the questions and comments coming through, and you can add your own. And then at the end, Ben and I will go through them and ask them, usually one at a time, but sometimes we have to pick them out. And if there are any left over, then Andy and Jon will be able to see them and possibly answer them after the end of the webinar. So cameras and microphones off, please. And I will turn mine off now and hand over to Ben, who will start the talk and start Andy. Thank you, everybody.
Well, thank you very much, Keith and Ben, for the kind invite for myself and Jon to talk to you this lunchtime about the Broadbalk experiment and various agroecological research that's been conducted on the experiment over the decades, and really pleased that Broadbalk's listed on the ECT list of sites now, together with Park Grass, which people are probably familiar with. So a bit of a double act today with me and Jon. I'll briefly introduce the experiment, some of the treatments, and I'll introduce this concept of how long-term treatments have created gradients, as we call it. And then I think the bulk of the presentation today will talk about the effects of these gradients that have been created in these in the Broadbalk long-term experiment, and their effect on the important agroecological functions and services. So I'll talk a little bit about net primary productivity, how agroecosystems regulate water and air, their role in mitigating or potentially exacerbating climate change. And then I'll hand over to Jon, who will talk a little bit more about support for biodiversity, which will include much more ecological aspects. So the Broadbalk winter wheat experiment, established in the autumn of 1843 by Laws and Gilbert, one of their so-called classical experiments established in the 1840s and 1850s, with the objective really looking at the effects of nutrient inputs on crop yields. And because at that point in history, there was certainly a lot of unknowns. And Broadbalk was essentially their experiment on winter cereal, winter wheat. So it's looking at inputs of nutrients either as inorganic fertilizers or as organic farmyard manure. Now, the experiments have three distinct phases to it, and I'll pick up this in a few moments' time when I show the long-term trends in crop yield. But we have an initial phase that lasted for a full 80 years, winter wheat being grown continuously, and the nutrient treatments applied in quite narrow strips along the full length of the field. So if we think of these strips as plots, there was only about 19 or 20 plots initially in what became the Broadbalk experiment. And since then, there's been a couple of modifications. There was one made in the 1920s, and that gave the scope to look at introducing a bare fallowing phase 1 year in 5, and I'll explain a little bit about why that came into operation. But that essentially split those strips into 5 sections. And then a major modification made in the autumn of 1967 for the 1968 crop and beyond. And now we have an experiment with many more plots, 188 in total. And we have different parts of the experiment where wheat is still being grown continuously, keeping the link back to the 1840s, but also where wheat's being grown in a crop rotation. Some of the important long-term nutrient input treatments include a control with no inputs at all, manured plots receiving 35 tonnes per hectare of farmyard manure on a wet basis, and a suite of various nitrogen input rates from 0 up to nearly 300 kilograms of nitrogen per hectare. The crops in the current rotation on that part of the experiment include winter oats and winter beans, but previously we've had spring beans on there, we've had potatoes and forage maize. And just to emphasise, it is a bit of a record breaker. It is cited in the Guinness Book of Records as the world's longest running scientific experiment, which is quite a fun thing to share. And we're obviously indebted to Laws and Gilbert for establishing it, and then all the people that have come after to maintain the experiment and all its resources. So application of long-term treatments has created gradients, as we term them. And because Broadbalk, the treatments are nutrient inputs, either as inorganic fertilizers or as organic farmyard manure, then the gradients largely in this experiment are ones of soil fertility and soil organic matter. So on the left-hand side, just as an example, we've got the top graph showing soil phosphorus and the bottom one showing soil potassium as well, time along the x-axis. And we can see that application of fertilizers containing phosphorus and potassium have created this wide range of different soil phosphorus and potassium contents, as we may imagine. And also soil organic matter, here expressed as soil organic carbon over time. And down the bottom of the graph here, we have the soil organic carbon stocks in the inorganically fertilized treatments on the experiment, largely in equilibrium. But we have increased soil organic carbon stocks where we've had long-term annual applications of farmyard manure. And a part of the experiment, small portion at the top of the field, about 0.2 of a hectare, that was actually taken out of cultivation in the 1880s and now supports natural regeneration woodland and permanent grassland, which we refer to with the slightly grandiose title of the wilderness, but also we can definitely spot soil organic carbon rates increase in there. Also important to note actually is that we have a small but measurable increase in soil organic carbon where nitrogen fertilizer has been applied, presumably because the greater crop yields from applying nitrogen are putting more organic residues into the soil through the root system. So we'll spend the rest of the presentation really talking about the effects of these gradients on the important functions, agroecosystem, agroecological functions and services. I'm going to talk a little bit about net primary productivity, how agroecosystems regulate water and air, and their role to mitigate climate change, and then hand over to John. So in terms of net primary production, it's an experiment that's testing the effects of treatments on winter wheat. So I'm now going to show 3 slides together, which will just take us through the history of the experiment and looking at the trends in crop yield. So all of these graphs, they have time along the x-axis and we have grain yield along the y-axis. So here we can see that we have the yields on this blue line at the bottom of the unfertilized, unmanured treatment at Rothamsted. Very low yields. But Laws and Gilbert were able to demonstrate convincingly and very rapidly that, as we may expect, inputs of nutrients, either as inorganic fertilizer or as the farmyard manure, increase yields, in fact, more than doubled yields. And in fact, there's very little difference in the yields between inorganic fertilizer and farmyard manure. Now, this part of the experiment, we had winter wheat being grown continuously year on year. And of course, at that point in history, we had the older varieties of cereal crops, very long strawed, but limits to how much they would yield in terms of their grain yield. As we approached the 1920s, it was noticed that the yields were beginning to decline, and it's thought that this was due to increased problems of weeds. If we think about that period of time, just after the First World War, shortages of labour on farms, and Rothamsted would have been no different, but also a time of great societal change as well, and migration of labour from countryside to city as well. So that actually sparked the first modification of Broadbalk. And what we did was, what those did at the time was to split the field into 5 sections, and that allowed for a bare fallowing 1 year in 5 to be inserted in rotation. And as we can see on the yield graphs, it did result in that recovery of yield. Still very similar patterns, farmyard manure and inorganic fertilizer. And we can see where a couple of other innovations came in, an introduction of a regular liming program to try and keep the pH about neutral, and introduction of chemical herbicides to manage weeds, but with an exception, because part of the field was deliberately kept free of chemical herbicides. And John will speak a lot more about that particular section of the experiment later on. And then we come to 1968, 1967, going into 1968, that season, major modification of the experiment, a splitting of the sections to give us 10 sections today. 5 of them still have wheat grown continuously, which keeps the link back to the 1840s, but the other 5 are in a crop rotation, which has changed over time. And the other important change made in 1967, 1968 was the shift towards the modern short straw varieties of cereals post-Green Revolution, which literally doubled yields. But the greatest yields, as one may expect, are for the winter wheat grown in a crop rotation, particularly the first crop grown after a break crop, typically a legume at the minute with winter wheat, and still very low yields on the unfertilized, unmanured control. Now talk a little bit about effects of long-term treatments on soil organic carbon and its subsequent effect on soil physical properties, and in particular soil structure. So the way that soils organize themselves into aggregates with a pore network, and of course it's the pore network that allows the water and the air to transmit through it. Now, colleagues of John and ours at Rothamsted in recent times have exploited X-ray computed tomography techniques. So this allows us to image at very high resolution soil structure in 3 dimensions. So exactly the same technology as you get in hospitals to image the body, so we can look at soil structure similarly. And modelers can take that image, that 3D image of soil structure, and can actually begin to quantify the pore size distribution and how the pores are connected to each other. And that's what the graph on the left is showing. For some of the treatments on Broadbalk, we have this measurement of connected porosity, essentially the proportion of pores that are all connected to each other. And we found increased connected porosity in the soils with greater soil organic carbon content, i.e. the ones that receive annual applications of farmyard manure, or that part of the experimental field that was taken out of cultivation and is now under long-term woodland and grassland. The diameter of the circles is proportional to the soil organic carbon content. But also, taking that soil structure, that pore network, that connected porosity, modelers can actually predict how water transmits through that. And that's what's shown on the y-axis here. K, hydraulic conductivity, essentially the rate that water will transmit through that pore system. And there's a nice relationship between the two. Greater connected porosity, greater ability for water to transmit through it. And also modelers can have a look at how pores relate in terms of their degree of saturation. So the proportion of pores that are water filled or those that are air filled as a soil drains. This middle graph here on the x-axis, we have matrix potential. So this is a kind of energy status of the soil water, if you like, moving from near saturation on the left to increasingly drained, drier conditions on the right. And on the y-axis, we've got the proportion of pores that are still water-filled or anoxic with no oxygen in them. And for any given kind of water status, water energy, we can see that those soils with greater soil organic carbon content have a much lower anoxic proportion. So more of these pores are air-filled and less are water-saturated compared to purely inorganically fertilized treatments, which have pores that remain water-filled at those same degrees of water energy status. And that has implications for various processes in the soil, not least denitrification. So denitrification can occur in wet soils, waterlogged soils, anaerobic soils, and that results in this conversion of nitrogen potentially to nitrous oxide, which of course is a potent greenhouse gas. And as part of this work, colleagues did look at nitrous oxide emissions and found a link between applications of nitrogen fertilizer and nitrogen lost as nitrous oxide. A little bit about agroecosystems' role in either mitigating or exacerbating climate change, and in particular here looking at carbon sequestration in the soil. On the left-hand side for Broadbalk, over time, we've got the increase in soil organic carbon as a stock, tonnes per hectare. And of course, it's increased through annual applications of farmyard manure. But the cautionary tale here really is that it does look like it's plateauing. And it's just to emphasize that soils probably inevitably have some kind of capacity to sequester carbon, which will be soil specific, and that initial very high rates of carbon sequestration presumably cannot continue into the very, very long term. But nonetheless, soils have an important role to play in sequestering carbon. This middle graph here shows carbon sequestration where that land was taken out of cultivation at the top of Broadbalk, and we can see corresponding increases in soil carbon over time and down the profile, not just in the topsoil as well. And colleagues have tried to estimate as well the amount of carbon that's locked in the above ground biomass. And in addition to the long-term treatments on the Broadbalk experiment, we've had a look at all the treatments on our classicals and long-term experiments a few years ago and tried to have a look at their carbon sequestration rates in relation to a theoretical threshold that was proposed as part of the Paris COP conference in 2015. This was a proposed threshold which would offset carbon emissions from all other sectors. And we can see how some of these management techniques may sequester at or above that threshold and some which are low. But that's not to say anything about the economic feasibility of applying some of these practices over very large areas of land. And the final slide from me before I hand over to John. I think those crop yield graphs that I introduced, they kind of demonstrate, I think, that Broadbalk has shown that we can sustain crop yields over the long term through inputs of nutrients, either as inorganic fertilizers or farmyard manure. But of course, I think the cautionary tale is that we must use these inputs judiciously. And today's chair might recognize this graph on the left-hand side. This is work of Keith on the Broadbalk experiment, and nicely demonstrating that as we increase nitrogen inputs rates in terms of their as a fertilizer, and also all the nitrogen that's added as part of the farmyard manure, So we get increased risk of losing that nitrogen as nitrate to drainage water. And there is a drainage ditch at Rothamsted at the Broadbalk experiment, which allows us to collect water that drains from the different treatments on Broadbalk. And finally, some more recent work by the students at Lancaster, co-supervised by Carly at Lancaster and John, and I had a little bit of a role here. He reanalyzed soil samples from the Broadbalk experiment from the sample archive, and he very nicely demonstrated increased accumulations of microplastics, particularly from treatments that receive annual inputs of fertilizer and manure, but also cautionary increases in the nil plot as well, because there's many other sources of micronutrients. And with that, I shall hand over to John now.
Thanks, Andy, and good afternoon, everybody. So I'm just going to share a few stories from what we call Section 8 on Broadbalk, which is quite special because it has all the same fertilizer treatments that Andy's been talking about, but it's had no herbicides. So we've got some quite interesting and special weed communities on that section. Thanks, Andy. Next slide. So these are some photos of the weeds, or we could call them arable plants for the purposes of this afternoon on Broadbalk. So on the photo on the left there, we're looking down the experiment, and we've got section 9 at the front in the foreground, which has herbicides. And then you can see the weedy plots behind that, which is section 8, which has never received any chemical herbicides. It used to be hand weeded before that. So what we've got are naturally assembled weed communities that are typical of the different levels of soil fertility, that are typical of that nitrogen gradient. So you can see some very common species that we're all familiar with, like mayweed and poppy. But then on the lower fertility plots, we have interesting species like shepherd's needle and a number of weeds that have become nationally rare, including shepherd's needle, also corn buttercup, corn cleavers, and Venus's looking glass. And in a couple of photos in the middle there, you see a contrast between what the plots look like. So on the bottom, you see the fertile plot full of blackgrass and poppies, and then the top middle photo shows the nil plot with much more stress tolerant weeds and a more open canopy. Thanks, Andy. Next slide. So the data set that's most valuable for looking at these patterns was started in 1991. We do have data on the weed communities going back before then, but in 1991 we started doing a standardized protocol annually, and we do this in early June, where we look at the presence of species in 25 0.1 meter squared quadrats and just record presence absence. So we have frequency data for each of the plots for a long time period, and they're the data that I'll mostly be talking about over the next few slides. You might recognize some of the people in this photo. There's Steve Moss on the left there and Peter Lutman on the right, who started and were responsible for collecting a lot of these data. Thank you, Andy. Next slide. So what are the general patterns we see on section 8 along these gradients? And I'll be talking about spatial gradients and temporal gradients. So the spatial gradient is our gradient of soil fertility, and you can think of it as a space for time substitution. So we've got some plots here which are typical in terms of the fertilizer inputs of the levels of nitrogen we used to apply maybe 50, 60 years ago. So we have weed communities that give us an indication of what weed communities were like in those environments. And what we see is the plots with the highest species richness, so we've got a number of species on the y-axis here, are the intermediate fertility plots. When you get to the high nitrogen plots, you tend to select for a smaller group of very competitive nitrophilous species like blackgrass. And then on the control and the low input plots, you're down to a smaller species pool of legumes and stress tolerant rudels. So the high species richness is found on the intermediate fertility plots around 96, 144 kilograms of nitrogen. Thanks, Andy. And then we can also identify groups of species that are preferentially found at different points along this gradient. So don't worry about the detail of this and the naming of the species. This was an American journal, is slightly odd. But what it does illustrate is you've got two groups of species on the left here. And again, you've got the nitrogen gradient on the x-axis and the frequency of these species on the y-axis. The two boxes on the left are our common weed species, and you can see that they are still found a lot in the high nitrogen plots. There are chickweeds and our blackgrasses and poppies and mayweeds that are our common weed flora today. The bottom right panel there are the more stress tolerant plants, and they're still very common in, for example, coastal environments. But the interesting group for me are that top right group, which tend to be preferentially found on the intermediate fertility plots. And this is why the species richness is higher there, because you've got this representative group, and that tends to be where you find the species that have become nationally rare. So it's where you find the most shepherd's needle, corn buttercup, and Venus's looking glass. And I would suggest that that's the habitat that we've kind of lost from our landscapes. That regularly disturbed environment with intermediate levels of fertility. So Broadbalk is giving us an insight into why we've seen the historical trends in weed populations and weed communities in the UK. Thanks, Andy. Next slide. So there, spatial trends. We can see how the nitrogen gradient is selecting for different functional types of weeds with different traits, and there's other work that goes into a bit more detail of that, and we've identified a rare weed trait syndrome based on those data. But what about temporal trends? Are we seeing weed communities shifting or drifting specifically with climate change? And we looked at that in this recent paper in Global Change Biology. And if you're not familiar with these ordination plots on the left, all it illustrates is that if a species has an arrow going to the right, it means it's become more frequent across the whole Broadbalk experiment since 1991. So we could say it's increasing with climate change. If the arrow goes to the left, it's decreasing with climate change. And the panel on the right just fits lines through that. So if a species has got a positive line going up, it's getting more frequent in our data sets. If a line is going down, it's becoming less frequent. And we are seeing these longer term trends or shifts in the relative frequency of different species on the experiment, which is interesting because it seems to imply that climate change is having an impact on weed communities. And specifically there, I picked out a couple of species that we're finding more frequently now than we did in the 1990s. Thanks, Andy. Next one. And we can start to explain these trends based on competitive dynamics. So this paper that is a few years old now was looking at a specific hypothesis for plant coexistence. And that hypothesis says that it's environmental variation, so seasonality, can support functionally equivalent species that are adapted to the same habitat, but one species might do better one season, another species might do better another season. And we identified these functionally equivalent pairs of species that had the same pattern of distribution across that nitrogen gradient, but responded differently to weather. And this is one example of that, where the corn buttercup prefers or is found more frequently when we have warm springs, which is what that middle panel is saying, compared to the shepherd's needle. And because the warm springs are becoming more frequent, we think this might be part of the explanation why we're seeing some species become more dominant on plots and other species less dominant. And again, that's an insight that we can get from these long-term data sets where we've got the consistent management treatments. Thanks, Andy. I recognise that time is getting on. So my last few slides, that's looking at the effect of the spatial and the temporal gradients on the weed community composition. What about the function of those weeds in terms of their competitive impact? So my last couple of slides are looking at weed competition. And again, this is the spatial gradient. So we've got the nitrogen gradient here. The green bars are from the plots that have herbicides, so there's no weeds. The yellow bars are where we've got weedy plots and we have no herbicides. Can you just click through the next couple of animations, Andy, please? So we see that typical nitrogen response curve where we use herbicides, but where we don't use herbicides, we don't see the added benefit of those fertilizers because all the extra nitrogen is being robbed by the weeds. And actually, the weeds are better at robbing the nitrogen at the high fertilizer levels because we've selected for those very competitive nitrophilous weeds. Interestingly, in the zero plots, we see higher yields in the weedy plot than the plot that receives herbicides because we have facilitation from the leguminous weed community. But the message to take away is that as we add more nitrogen fertilizer, the weeds become more competitive and we lose proportionally more of the yield to the weeds. Next one, thanks, Andy. And the other thing we found out recently as well is, as well as seeing a shift in the weed competition along the nitrogen gradient, we're also seeing the weeds become more competitive with time. And in this paper that I've already pointed you to, we were able to show that the increased competitiveness of weeds was a combination of environmental change, so the season becoming warmer. So on the left there, you see that increased competition with time. So this is the yield difference, the difference between the weedy plot and the weed-free plot. So it's how much yield the weeds are robbing. They're becoming more competitive. They're taking more yield. That's because they prefer the warmer autumns and winters. So it's shifting the competitive balance. But also, secondly, on the right-hand graph, we've changed the cultivars that we're growing and we've changed the management, and our cultivars have now become less competitive. So Andy, next slide. So this is some photographs, a time series of the cultivars we've grown on section 8 since we started using herbicides on the rest of the experiment. And again, you can see they're getting shorter. So it goes from top left 1960s to bottom right 2015-16. So in the paper, we talk about a triple whammy in terms of weed competition. By adding more nitrogen fertilizers, we're selecting for nitrophilous competitive weeds. So we've shifted the balance in favor of the weeds. By growing shorter, high-yielding cultivars, we've also made the weeds more competitive because we've reduced the shading of the crop canopy. And thirdly, climate change and warmer autumns and winters have again shifted the balance in favor of the weeds. So we've stacked the decks, if you like, in favor of the weeds. As long as herbicides keep working, that's fine. But if you then put that in the context of herbicide resistance, the potential problem from weeds is becoming more serious. And again, we've gained insights into that from Broadbalk. Okay, thanks, Andy. I think I've nearly finished. The last couple of slides are to put a bit of balance, because obviously I know about all the weed work in detail, but I just wanted to highlight another couple of areas where work has been going on that might be of interest to people. One is on the soil microbiology. So there are a series of papers now by Andy Neal that look at the effect of the treatments on both the assembly of the soil microbiome and also its function. So this particular graph is looking at functional genes. And again, I'm not going to talk in detail about it, but just point you towards that work. And the second one, again, just for balance, is to highlight some of the work that's been done on earthworms. Thanks, Andy. Next one. Not to talk about it in detail, but just to make 2 points. One is, yes, we do have some work. You see this paper's quite old now. We do have some work on other parts of the system, other trophic levels and other taxa on Broadbalk, but there's still lots of potential. There's lots more we could do on the ecology of Broadbalk, and particularly the interactions of these different taxa and trophic levels. And with that, I think we're going to draw things to a close just with a final couple of slides. If you can just click through that. Thanks, Andy. So this summarises where we're going with Broadbalk, with a couple of photos on the right illustrating how things have changed. But traditionally, the experiment has been used to look at soil questions, soil chemistry questions, agronomy questions. But because of the gradients that the treatments have now set up, they're really valuable for looking at the behaviour of ecological systems. And the implication of that for ecosystem services and natural capital. So it continues to be an absolutely unique and valuable resource for looking at agroecological questions and the contribution of natural capital and ecosystem services to supporting sustainable production. And with that, I think there's just an acknowledgement slide, and then we can move on to questions and discussions. But on Andy's behalf as well, I thank you very much for your attention. Thanks.
Thanks both. Andy, is it worth just mentioning that Rothamsted's always been keen on collaborative work? John sort of hinted at it, but
yes.
People approach you for to work on the experiments or the stored samples or whatever. Do you want to just say a quick word about that?
Yes, yeah, and it's linked to this slight mouthful of national bioscience research infrastructure. That's the term that our main strategic funders, BBSRC, refer to as, and the clue is in the title there. They do see this as like a national research platform, really. So we do need to demonstrate that it's not just supporting Rothamsted science, that it is being used in particular by UK biosciences, covering the full biosciences, and increasingly international. And we do that. We do get a lot of requests to either measure things directly in the field, certain traits, or to collect samples of crops or soils from the field for analysis. The sample archive that some of you no doubt will have seen, and the data sets as well is the other real component. And I think the experiment's still in the field. The sample archive, the data sets, does make Rothamsted unique in these long-term experiments. So yeah, we're always very happy for them to support new science.